Okay. So now I will introduce our first speaker, Ari Mailhos, who will be presenting his talk on Pappus diversity in Compositae.
Thank you, Abby.
Thank
I'll
you.
share my screen now.
Yes.
Let's see. There we go. Yeah, looking good.
Looking good, yes.
Do you guys see my mouse when I move it?
Yes, we do.
Awesome. Then let's begin.
Okay.
Well, good afternoon, morning, night, whichever applies to you, everyone. Thanks for being here. Um, like Abby mentioned, I'm going to be giving a talk that is part of the project I'm carrying out as part of my PhD thesis that I am doing under the supervision of Mauricio Bonifacino here. And it centers around Pappus morphology and diversity in the Asteraceae, right? So, as part of this talk, I'm going to be giving some general background on the Pappus as a structure in general, and then we'll be going into some detail into the morphology and anatomy of the Pappus, showing some of the main examples we can find within the family. And then we're going to cover the functional diversity that we can see within the Pappus in different groups of the Asteraceae. And lastly, we're going to be talking briefly about what correlations may occur between the morphology of the Pappus and its function and other structures within the capitulum of a plant. So I think everyone here knows this pretty well, know the basic structure of a capitulum, but this is just to say that the Pappus as a structure is one of the most distinguishing characteristics of the Compositae, right? It's this highly modified structure derived from the calyx that is usually persistent in the fruits of these plants. And it has played a key role in the evolution of this family, right? It is believed to have been one of the main reasons for its evolutionary success. For instance, it's what it allowed early combs in the evolution to disperse long distances across continents and colonize and radiate in new habitats and places around the world. So yeah, we basically have the Pappus to thank for that. And fossil evidence and other studies have shown that the Pappus as a structure, like we know it nowadays, arose pretty early in the evolution of this family. Now to briefly touch this point, because this is like the age-old question that was the reason for many debates, especially throughout the 19th century, right? Botanists at that time were really couldn't agree if the Pappus were some kind of specialized trichomes or if it was in fact a modified calyx. And even nowadays, you sometimes hear people casting doubt on its nature, right? But I can confidently say that we have pretty solid evidence that the Pappus is in fact derived from the calyx. There have been some studies in the past few decades that have shown that the genes that regulate the expression of the Pappus are homologous to genes regulating calyx expression in other plants. So that's a pretty solid evidence for its origin. Also, I should mention that Pappus elements in many species have vascular bundles inside within them, which we wouldn't find if it were a structure derived from a trichome, right? So that's just so we're all on the same page here. But I want to say that that is not to say that within the astounding diversity we can see within this family, who knows if perhaps some weird comb somewhere does have a structure resembling a Pappus that is actually of a different origin, right? I mean, and there are some possible candidates that have been pointed that where that might be the case, but I haven't been able to find any concrete proof of that, but it's certainly something to bear in mind, okay? So now to give you an overview of the classification, morphological classification of the Pappus, and we can't ignore the work that James Small carried out nearly, well, bit over 100 years ago, where in his series of papers are about the morphology of the Compositae, he dedicated one specifically to the Pappus, where he compiled a lot of information that was available at the time, together with his own observations, and he provided a pretty exhaustive classification proposal for the Pappus across the whole Compositae, right? And this established like the basis for Pappus classification that we still use nowadays. So basically, uh, Small recognized four big groups of Pappus morphologies that can be characterized as setose Pappus, which is composed of elements that are more or less thin and elongated with a more or less circular section. Then we have the aristate Pappus, which is composed of awns. Then there is the palliaceous Pappus, which is composed of scales or flattened elements of varying size and consistency. And lastly, there are the coroniform Pappuses, which are like a small crown around the top of the cypselli. And they can be like a whole membrane, or they can be more or less differentiated into tiny scales. And there can also be combinations among these types of elements, as we will see now. But I want to also point out that within each of these types, as we can see in this illustration, there are different kinds of ornamentations that we can find associated with these elements, right? For instance, in the setose, we have these ones here, which are typically called the plumose bristles, for instance, or these ones which are barbellate bristles or scarped bristles, etc. And so on for the aristate, many of which have these retrose projections, but they can also be antrose, or they can be lacking entirely, and also in the palliaceous, as you can see there. Also, beyond the individual type of the elements which compose the Pappus, we can also classify the Pappus according to if it's composed of a single type of element, like the drawing here on the left, or multiple distinct types of elements in a single cypsela or floret. For instance, here you can see that at the base there is a small series of scale-like elements, and then we have these longer bristle-like elements. So when we find more than one type of element, we say that that Pappus is heteromorphic. When there is a single type of element, we call it homomorphic. Also, we can classify the Pappus according to the number of series in which these elements are arranged, right? From one series to multi-series, it can vary greatly, the number. Something to bear in mind, when we say that a Pappus is heteromorphic, we have to specify if we are referring to a single floret or we are saying heteromorphic within the capitulum, right? In this previous example, it is heteromorphic in a single floret, right? A single floret has two distinct types of Pappus elements. But in some species, we can find that different florets in the same capitulum have different types of Pappus. And we have this example here in Erignois in the Sicori, where the florets positioned more to the margin of the capitulum have this coroniform Pappus, and this one on the right is present on the more central florets in the capitulum. And in the middle, we have some intermediate forms between one and the other. So when we use the term heteromorphic, we have to be precise and say if it is heteromorphic in a floret or within the capitulum. Also, Pappus elements may be fused between each other, or they may be free. And that is also another characteristic we can use to describe the Pappus in general, right? And well, finally, I have to mention that obviously many comps lack a Pappus completely. There are some tribes that are characterized by this. For instance, many Anthemidae don't have a Pappus, the Calendulae, for example, and some others. And then some specific genera within other types that typically have a Pappus suddenly lack a Pappus, like this Pamphalea in the Nosovii. So a while back, I had the opportunity to visit the Smithsonian Herbarium and Museum, and I spent a couple of weeks gathering samples from the of Pappus from the herbarium there, and taking a great deal number of scanning electron microscopy images, which I'm going to use now to show you some of the morphological diversity we can see within the family, right? To illustrate some of these different types of Pappus I've just mentioned. So for instance, here we can see what is typically called the capillary bristles, right? This is like a bristle type element, but it is composed of very few cells in cross section, right? So here we can see this in this lactuca. There are only like 2 to 3 cells in diameter. Very, very thin, delicate elements. In contrast, we have these, which are the scabrous bristles, which are perhaps, don't quote me on this, but the most common type of Pappus across the family, right? Many species and many genera across most of the tribes show this kind of Pappus, which are composed of bristles, which have projections that are at most equal to the diameter of the bristle, but not much longer than that. And I also want to point out in this image I chose here to illustrate this, that the shape of the tips of these projections may also vary, right, in different parts of the same element. Like typically, in some species, the lower projections are more acute, and at the apex, they become more obtuse or rounded. And this is something that is very useful to separate species and genera. Like Robinson, in his treatment for the Eupatory, paid close attention to this and showed that yes, it can be useful to separate taxa. Then we have barbellate bristles, where the projections of the bristle are longer than the diameter of the bristle itself. And the projections here are typically rigid, and there is great diversity in how they are arranged. They can be arranged like only on one side of the bristle or all around, like we can see in this image. But it is not much more complex than that. And finally, we have the plumose bristles, where the projections are many times longer than the diameter of the bristle, and they tend to be very thin projections and delicate, right? Typically, Pappus that have these types of projections are clearly dispersed by the wind. Just to show you some more examples of an heteromorphic Pappus, like this is a very contrasting example in Dicoma, where we can see these scales in the innermost series of the floret and these scabrous bristles toward the exterior, right? But we can see that two very different elements can coexist in a single Pappus. And then, well, just to show you some other examples of other types, here is the Sclectendalia Pappus, which is composed of these very thin, long, elongated scales. But we can also have scales that are more rigid in consistency, like in this Eupatory showing here. And these ones are also shorter and more or less approaching what could also be considered a coroniform Pappus, which are typically a bit shorter. And then just to show you that there are many intermediate forms within these basic types. Here is an example in the Vernoni, which has these very thin elongated scales, but in literature, they are sometimes referred to as flattened bristles. So there is like no consensus on what they should be called. And notice also their three-dimensional arrangement, right? How they form like this spiral, which is pretty interesting. Not what you typically find in most species. Then we have the awns. These are pretty common to find as well, like in these bidens. And just to show you some more examples of heteromorphic papuses, here awns and scales coexisting in the same akin. And lastly, a coroniform pappus, which, like I mentioned, is typically found or formed of a short structure with more or less lacerate edges, margins, where we can sometimes see the individual scales that conform this structure, or they can be more completely fused, and we can't really tell one apart from the other. And when we look inside this pappus, here is a sample of some of these different types, right, in cross section. And other characters that we may look at here to provide a characterization of the elements is the type of wall of cell walls present, like we can see here in this porophyllum and the telesperma and bidens. We have some sclerenchymatose cells with very thickened walls, right? In contrast, for example, to the Senecio or Noticastrum. And also, like I mentioned earlier, some Pappus elements are vascularized, and we can see vascular bundles. Like, for example, in this Galinsoga, here, this would represent a vascular bundle. In Telesperma, I think around here, that also corresponds to another bundle. So these are all characteristics that we can use to provide a description of Pappus and separate genera and species. But all of this diversity has led to a great number of terminology used to describe these elements beyond what Small initially proposed. So nowadays, the situation is rather complex because one of the main issues that inspire this project we're working on is that we saw that there was not a consistent use of terminology across tribes and sometimes even within tribes or within a single flora or taxonomic treatment. So, because even though Small's treatment of the Pappus was very exhaustive and very descriptive and very well illustrated, still to date, we do not have a concise and precise definition for most of these terms and these elements. Like some have been proposed within a specific group, a specific genera, or a specific tribe, but there has not been a classification proposal that attempts to provide a standardization for all of these descriptive terms that englobes the whole family, really. So that is more or less what we are attempting to do here. And so yeah, we are looking at Pappus examples from across the family, trying to sample as many tribes as possible, and doing an exhaustive revision of the literature to try and compile all of these terms that are mostly used. And based on all of that and detailed morphological and anatomical studies, we're going to try to provide a specific definitions and accurate definitions for the terms that we consider more apt to describe the Pappus. We hope can serve as a basis going forward for the description of the Pappus. So that's what I had to say about the morphology, more or less. And now I'm going to be talking a bit about the function of the role the Pappus plays in the life of these plants. So when one thinks of the role of the Pappus, I think, at least in my case, where one's mind goes first to is an example like we have here in this image of a Hypocheires, right? Of a typical bristly Pappus that is easily dispersed by the wind, right? We've all taken a dandelion and blown on it and make all of the stella blow away. So I think that's a very present image in our brain. And what we have found looking at the literature at that most studies that focus on the pappus and its ecological role tend to focus on this aspect, right? It's dispersal, which can be usually, yeah, by the wind, but we can also see adaptations of the pappus that are more geared towards dispersal by animals, like in these bidens, where those rigid projections get entangled in the fur of animals, right? Yet another example of this is this Adenostema in the Eupatory, which has a very particular type of pappus, which is composed of some few ons, which have some glandular trichomes densely covering the apex of the on, and they secrete a very sticky substance. So when you brush against them, they get stuck all over you. So it is quite an effective means of dispersal that these species have. But beyond the role of the pappus as an agent that facilitated the dispersal of the fruits and seeds of these plants, there hasn't been much else looking at other possibilities. That's not to say that there hasn't been anything at all, but I'm going to shine a light a bit on some of these other stranger cases. For example, this is a very particular case. This is the genus Fulcaldea in the Barnardici. This is a South American genus, I think composed of only 2 species. These plants have capitula with a single flower on them, and the corollas are not too visible, not too yeah, not too. They are colorful, but it's just a single corolla, right? So the most attractive feature in the capitula of these plants is actually the pappus, which when the flower opens and matures, the pappus is already really well developed and it's very colorful. And these plants are believed to be pollinated by hummingbirds, hence the reddish coloration we can see there. So in this genus, the pappus actually serves a function to attract the pollinators of this plant, right? Which is something that one wouldn't expect when thinking about a pappus. And then we come across things like this. Look here how in these capitula that are developing and the florets haven't opened yet, we can see that the pappus is already pretty well developed and how it fills in the spaces between flowers and almost seems to cover them, right? And look at this one also in the Vernoni again. This is a maturing capitula where the filaries have already opened at the top, but when we look inside, all we can see practically is pappus, covering all the developing florets, right? So things like this led Stussy and Garver in 1996 to write a short paper where they shined a light on the defensive role that the pappus appears to be fulfilling across Compositae, right? They say that yeah, the pappus beyond an agent facilitating dispersal can also act as a physical barrier against predation from insects mostly. So in their analysis, they mentioned that an insect typically would want to access the ovaries or the fruits in a capitulum, which are like the most nutrient-rich structures present there. And they can access this part of the capitulum through 2 ways. Through the lateral way, right, where the main barrier would be the filaries and the involucre, or they can access from the top, where the main barrier would be, well, the corollas themselves, but also the pappus that is present there. So when we take that into perspective and look back at this plant I was showing you here, right, we can see how it kind of makes sense, right? The pappus is completely filling in those gaps between florets and really creating a barrier, a physical barrier that small insects would find difficult to overcome. And it's also, Tracy and Garver also point out that most of the many pappus elements have these projections that point towards the top, right? So thinking of an insect that is coming down, this would create a stronger barrier against them. So when I first read that, yeah, it kind of opened my mind a bit about this. And then I started to pay more attention whenever I came across a comp in nature in general. And I started to see that, yeah, in fact, one can see this pattern repeating across many different genera and tribes. Right here, we have another example in the Abbey where you can see the exact same thing. The pappus covering completely all the developing florets. And this is a very neat example because here we're looking at the staminate, or functionally staminate capitula of a Baccharis, right? So these capitula do not produce fertile cypseli. So they would not need a pappus to disperse the fruits. So then why are we seeing all of these pappus, really well-developed pappus, that is already present and covering all these florets? The main reason that it might be there is, in fact, yeah, to provide an additional layer of protection for those florets. And then there is an even more extreme example. I don't know if you've seen images or in person this plant before. This is Stiftia, where before the florets mature, the pappus form a really dense, compact cluster around the florets. Right here, we are seeing the immature capitula, and all of these that we are seeing here are pappus bristles, very tightly pressed against one another, forming a really, what one would imagine, impenetrable barrier to an insect if it wanted to access the florets inside. And when I looked at those bristles under the scanning microscope, I found that, yeah, on the outside, well, first of all, they are very robust in comparison to typical bristles. Like they are really large in diameter and in length. And they have all of this great number of projections on their outward facing surface, whereas they are completely smooth on their inner surface, right? So all of these densely clumped together, one would imagine that, yeah, it probably does make a pretty effective barrier. And then I found that it is not only Stiftia that does this. For example, here, well, I show you Famatinanthus, which is kind of a bit of a rock star right now. But we can see that, yeah, it is doing the exact same thing in the early stages of the development of the capitulum, right? So it's pretty curious. And what's also interesting is that in the Caliceraceae, most species, the sepals, also appear to serve this sort of function. They are typically scale-like and rigid, and they seem to form this like barrier preventing insects from accessing the ovaries and fruits, right? So Strucy and Garver, they position that the pappus initially in the Compositae may have been specifically fulfilling that same role, and that its role as a dispersal agent may have come later. However, I've also noticed that not all comps do this, right? In many Sicori I come across, I did my master's degree studying a group of South American hypochiris, also in the Sicori. And in those species, you practically do not see the pappus until the capitulum is fruiting, right? When it's maturing and in anthesis, like the pappus is very inconspicuous and it is practically hidden between florets and it's not really well developed. Like the pappus only really matures when the fruits are maturing. So yeah, this is just to show you that, yeah, there appear to be some different patterns occurring in different groups within the family, and that is also something that I am looking into, right, to see if there are in fact any evidence to uncover any phylogenetic patterns surrounding this development of the pappus in relation to other parts of the capitulum. And Stussy and Garver, in their paper, they also include this sentence, which says that if the Pappus in the Compositae really does have a defensive adaptive role, then one should be able to see correlations with other structures in heads of the family, right? So they think that they talk mostly of the Pappus in relation to the Paley, for example, and they say that within lineages that, for example, tribes that have genera with and without Paley, that maybe one can see a sort of compensation mechanism going on where, because the paley also serve a protecting function, right? The paley typically envelop the florets and cover the ovaries and fruits. So in species where the paley are well developed, those species would not need to have a pappus that is fulfilling a defensive role because that role is being fulfilled by the paley and vice versa, right? And they mention this, but they say that this is something that should be explored and that is something that is likely happening, but there is no concrete evidence for that. And then a few years back, in a paper by Mauricio and Vicky Funk, where they studied this group of the Asteri in South America, which is a group of species that have paleys in a tribe that typically doesn't have them. And they saw evidence that seemed to support Stussy and Garver's hypothesis, right? In this paper, they saw that in species, there were associations between the degree of the development of the pappus, like how large it was, what kind of ornamentations it had, and the presence or absence of palea in those capitula. And there seem to be also some other correlations between the number of florets in a capitulum as well. Right, so there seems to be some evidence that, yeah, in fact, there are some correlations, but this still needs to be looked into in more detail. And I think also a pretty good example supporting this is that in groups that have like the Elianthe, where a great deal of genera in that tribe have a really robust paley, we can see that the pappus here in this species is typically composed of a small short scales or awns or some small coroniform structures that one does not imagine are fulfilling a really effective defensive role. And one can think that, yeah, in these plants, the paley are the main structure serving as a defense. So these are all avenues that we are exploring and particularly looking into, trying to find these kinds of groups and see correlations into them. Like I mentioned, it's specifically interesting for the purposes of this to find examples of species of genera that either have paley in a tribe that on a group that typically lacks them, or vice versa, right? And see how that affects the development of other structures. So far, I've managed to gather samples of a great deal number of tribes within the family, like most of them. I've sampled at least one genera from. And this is all in part, yeah, to carry out those morphological analyses to be able to propose a classification and a standardized terminology for the classification of the pappus and well, to look into, yeah, its relationship with other structures. But this is an ongoing work, a work in progress. I still have a bit of work ahead of myself, but just to show you something of what I'm doing, what we are doing together with Mauricio. And yeah, just to let you know that if you know of any interesting, weird, or cool pabbuses, or you come across them that you think that might be of interest for this project, please get in touch. If you have images or illustrations, or just to let me know this genera has this type of strange pappus, more than welcome. So there's my email address in case you might want to contact me. And well, thank you very much for listening.
Very interesting talk. Thank you, Ari. So do
Okay.
people have any questions? I have a question. Oh, did you have something, Marissa? Go ahead.
I'll go after you. It's okay.
So at the start, you talked about that there's a possibility that there might be pappus that are actually modified trichomes. So what would you look for in a pappus element to think, okay, this is maybe something that should be investigated further with, I don't know, genetic mechanisms or something like that?
Yeah, it's a complex issue. There are some, I don't remember the name. There is one I come across where literally like what is described as a pappus, there are like some really small structures at the top of the cypsela that are like only a couple cells in length, that those look really suspicious to me, for example. And also in the Elianthe, there are some that seem to or look like this would not be a trichome, but there are some that might be projections of the cypsela walls that also I think might be happening in some other groups. But I sadly I don't think there's any like specific thing that would be like a very clear giveaway, right? I think we just have to look at a list of potential suspects and well, hopefully someday someone will look into the genetic part of that and be able to confirm. But yeah, it's not as easy as it's might
Yeah,
be.
definitely.
Yeah. Mauricio, do you wanted to say something? Yeah.
Yeah, I wanted to make a comment, Marisa, if you allow me. This is connected to this question that Abby had. Abby, you remember that we had this genus Berroa in the Nephali that
Yeah.
has a strange situation. Perhaps you want to comment that example because that's a good example and something that could lead you off track.
Yeah, I don't have any good images of that one, but yeah, in Berroa, they're like it's what looks like an heteromorphic pappus, right? Where but when it falls like one part, the innermost part of it, if the seed is deciduous, it falls away pretty easily. And then there are these like some few very thin and long bristle-like structures that remain attached to the cypselae. So that might be like a mixture between a true pappus and some other accessory structure, right? Of trichome origin. Yeah.
Thank you.
Yes, Marisa.
Hi. Hi. Yeah. Really interesting talk. So you mentioned the possible defensive mechanisms of the pappus, especially when they're really dense or kind of covering most of the florets. I'm curious, in instances where there's like double, triple, or quadruple like series of pappus, are you aware of any experiments, maybe like gene knockouts or whatnot, to see like what happens like if you remove the pappus or like some series of pappus? Like, is there some change to how the plants are defended from insects? Do you know of any gene knockout studies for any of this?
Yeah, I am not aware of any studies that have attempted to do that, sadly. But yeah, that would be, I think, really interesting to do. And it will be the well to definitely prove that it has that defensive role for sure. The studies I've seen where they attempt to do gene knockouts are the ones where they try to ascertain the origin of the pappus, like as a modified calyx, right? There is one that they did for Taraxacum and in Gerbera, in the Mutisi. Those are the ones I think that have been studied from that point of view. But looking into this defensive role, I'm not aware of anything that has done that.
There are no further questions. Let's thank Ari again for a really nice talk. And then we can move to our second talk by Joseph Kleinkopf. And he will be telling us about the evolution of alpine fleabanes, Erigeron, in North America.
All right, thank you. Can you see this and hear me?
Yes, yes to both.
Okay, perfect. Thank you. Well, I'll start by saying thanks for the invite. Colin and Isaac invited me late in the spring. And yeah, it was a great opportunity. I didn't realize there was a whole seminar series just for the Compositae. So yeah, excited to be here and share a little bit of the work I've been doing. So yeah, my name is Joseph Kleinkopf. I'm a PhD candidate in the Marks Lab at Cornell. And I'm going to come at this kind of from the biome perspective, because I'm really excited about the Alpine in our temperate North America region. And then so I'm not as familiar with the taxonomy of this group, so I'm still learning that, but that's kind of the background to this. Okay, so here's a picture of Wheeler Peak. This is the highest point in the state of New Mexico in the US, which is where I started doing a lot of my alpine work. And I really like this picture because you can see this tree line, which starts at around 12,000 feet elevation in New Mexico, above which are almost these barren looking mountaintops, and that's the Alpine. And of course, they're not barren. There's lots and lots of stuff growing up there, but they're experiencing these really extreme environments, these short summers, only 2 to 3 months long, intense UV radiation, strong winds. There was one trip where we couldn't even stay up there. We had to actually come down because it was so windy. And so there's lots of extreme stuff going on up here in the summer months. And then the winter months look like this, where actually when there's a lot of snow, the plants are protected. And where there isn't snow, they're experiencing these extreme cold temperatures. And Alpine is also really interesting from a biogeographical perspective. So on the right is a map of North America, and the bright pink is Alpine. You can see how fragmented it is across the landscape. And so, you look at these mountains, like the one on the left, they look like little islands coming out of a sea of trees. And so they become really interesting from kind of a island biogeography perspective, which is also why I'm interested in them. And so for the past several years, I've been up in the Alpine, just trying to document baseline biodiversity, because we know, what plants can live in the Alpine, but we don't really know what a community looks like, at least in fine detail. So we were characterizing individual mountain communities. I was in the Southern Rockies, but I had collaborators across the Northern and Middle Rocky Mountains, and then in the Cascade Mountains. And in that time, we documented close to 800 species of flowering plants from around 35 families. So it's really quite diverse, even though it's so extreme, and these communities are so fragmented. And so my question, was just, where do alpine plants come from? They're stuck at the tops of these mountains. Some are generalist species that can dip below tree line, but there are also quite a few that are only, at the very top. So where are these coming from? And there are a couple of hypotheses. One is the Arctic hypothesis, which is one Darwin posited in his On the Origin of Species. And he basically suggested that alpine plants originate in the Arctic somewhere, and then during glacial periods are migrating kind of down these mountain areas because alpine is lower. And then during interglacials, they migrate back up the mountain or back northward. And so you get these pockets of alpine Arctic taxa. Or alpine plants could just come from the lowland. They just migrate upward. The gradient of environments is a little bit more, is steeper that way. So it might be harder, but that doesn't mean it's impossible. And so these are the two competing hypotheses. And of course, I'm not the only one who's interested in this kind of thing. There was a recent study on Saxifraga that basically just shows that there are more upward migrations from lowland into alpine than there are alpine to alpine migration events. So this lowland hypothesis is supported in this group. And then this other really cool paper by Ding et al., 2025, that asked a similar question, but they just pulled a ton of GenBank data and built reconstructed trees for these groups, and then did a similar thing looking at distinct mountain regions across kind of the temperate parts of our world. And what they find is that the alpine floras are kind of the result of a combination of migration, right, colonization from other alpine regions, upward migration from the lowlands, and cladogenesis, so sympatric or allopatric speciation. And what's interesting is in North America, these three processes seem to play almost equally important roles. So you have colonization, cladogenesis, and expansion playing, like they're all equally important. And that got me thinking about the relative importance of these roles in these distinct geographic regions of North America, because as we know, North America is pretty complex geologically. So I decided to take a similar approach, but just focusing on North America here. And I broke North America into kind of five regions broadly defined by their floristic kind of affinities. So these include the Arctic, and then the Alpine. So they have kind of similar floristic affinities. The Boreal, which is this northern area that has trees and mountains, but isn't the Arctic. And then the temperate mountains here, kind of in the central USA area. And I know there's a lot of mountain systems here that are really unique and distinct, but for my question, it kind of made sense to group them all together. And then the southern mountains. Here I'm showing just the highlands of Mexico, but this region, I've kind of encompassed all of Mexico, the Central America region, and South America. But I think it would be really cool to do this again with different, regions and different classifications in order to get at some different questions. And then anything that didn't fit into kind of these mountain regions, I put into the lowlands. And then the basic idea is to reconstruct a phylogenetic tree. So for this case, Erigeron, download a whole bunch of GBIF occurrence data and overlay that with my shape files. And then using that, assign species to their biomes. So we could do alpine, non-alpine, or both, and then to their distinct regions. And the idea is to use ancestral character state mapping to go back in time and assign what we think these nodes could have been. And then from there, we can count the number of times we find an alpine to alpine dispersal event, or maybe an upslope biome shift event, or alpine speciation events. And so that's the general kind of framework that I'm working with. And so why Erigeron? On its face, it's a really cool group because it's really speciose. There's a ton of species in this group, 400-ish. Some people say 500. And it evolved around 8 million years ago. So the Earth was getting colder, but it hadn't reached kind of the peak coldness yet. And for me, I wanted to find something of North American origin because there are a lot of species in the Alpine that have whose groups have a primary origin in Eurasia somewhere. But I thought it'd be nice to look at a North American group. And then there's a diversity of regions, habitats, and range sizes in this group. So we have taxa that are narrow endemics that are found in the Alpine or Arctic, some taxa that are narrow endemics that are non-alpine, and there's a lot more than what I'm showing here. There's some really widespread high latitude or high elevation species, and then some widespread lowland species that are found kind of all over the lowlands. Some of these are even invasive in other parts of the world. And so we have this huge diversity of taxa here. But one issue, of course, with a lot of these interesting groups is that there's not a really good tree. So this was the most recent kind of concerted effort to try to reconstruct phylogenetic relationships in this group. And as you can see, there's a lot of polytomies. There's a lot of relationships that are not resolved. So my goal was then to kind of work on this tree and then use it to answer these questions that I was interested in. And so the methods, pretty straightforward, I think, to sample and extract DNA from as many species as possible, using primarily herbarium specimens, but also silica dried tissue samples that we collected. And we were able to collect a lot of tissue samples from a lot of specimens. And this was in large part thanks to so many herbarium folks who are working all the time to help people like me out and collect samples. And we were able to extract DNA for quite a number of them. And then we sequenced single copy loci from across the genome. We used the 353 base set, but we also designed a custom base set of 1,030 putatively single copy loci. And then we added 5 cold response genes that we thought could be interesting to look at some other time. And then we reconstructed phylogenetic relationships using a pretty standard workflow. But after all the filtering and all the sequencing error with older specimens, we were left with around 206 actual in-group taxa. And then this is something we're still working on, is the actual modeling the biome evolution biogeography. So I'm going to have to do some hand-waving biogeography, but hopefully it'll be interesting and give you some insights into what's going on in this group. So as we probably already knew, Erigeron is a complex group, but here's a phylogenetic tree, and these are local posterior probabilities plotted on the branches. And so we have pretty good local posterior probabilities on a lot of the branches within this tree. And I know you can't read the text, but I'm going to zoom in on a couple of places that I think are interesting. There are some areas where this local PP is actually pretty low, and I think that's a lot in part due to hybridization. But yeah, when we look at the gene concordance factors, we find a lot of paraphily, and this isn't a surprise. There's a lot of species that have arisen over the course of 8 million years, and we know there's hybridization and integration, and there's probably some stuff at the genome level going on. But there are a lot of clades that we do expect to see actually come out in the phylogeny itself. So here's just an example. Erigeron section Tridactylia is this group defined by morphology, but they come out as this monophyletic group. And so we have some confidence that this tree shows at least some of the relationship we're expecting to see. As far as I think the classification is really interesting, I'm still kind of wrapping my head around a lot of this stuff. But the first thing I wanted to note is that Erigeron is paraphyletic, so I'm Sensulato Erigeron. But we knew this already from the previous phylogeny, and we see it again here. So that's a good thing. There are several genera that come out from within Erigeron. We have Neha, Hysterionica, and Apopyros. These are South American taxa. And I wasn't able to sequence Leptostelma or Darwiniothamnus, but those are also expected to be kind of coming out from within Erigeron. And then in North America, we have Aphanistephus, which comes out also as kind of this within Erigeron. And so that's good to see. But I think it's great that interesting to see that we're seeing that still. Another interesting thing is somebody gave a recent talk on Orotrophium, and I didn't include this in my phylogeny at the time because I was working off of the 2008 classification of Erigeron. But they showed that Orotrophium herdopilosum and nevadense come out in this group with 2 Erigeron species, Erigeron ecuadoriensis. And so you can see they look not the same, but they look pretty similar here on the left. And so it'd be interesting to include a couple of these species in this phylogeny and just see where they come out. Because that'd be another group that we didn't think about that makes this group paraphyletic. There are a couple of taxa that we included that are currently called Erigeron breweri, and Erigeron columnaris that actually come out with the outgroup taxa. Erigeron breweri looks like an Erigeron to me, so I'm not really sure how or why that would be coming out as an outgroup taxon. I looked at the specimen itself. It looked like an Erigeron breweri. But then this columnaris species, the right picture, has some characteristics of like a Caniza or something else that make it less likely to be an Erigeron. And so it'd be interesting to, again, look at all of these species that we have and put this into a phylogeny using the sequence data that we're looking at and see if we can reconcile morphology and taxonomy. Nisam in 2008 suggested a few alternatives to the classification of this group. So he suggested that we could do a clade-based taxonomy. So we could have Erigeron, but then it'd become a smaller group. We could add new genera describing some of these monophyletic groups within Sensulato currently. We could enlarge Erigeron to just include everything. I don't know. And then we could keep Erigeron as a paraphyletic group, but then recognize some of the monophyletic taxa that come out from within it, which sounded good to me. But we're also seeing that there are some taxa like Neha coming out in different areas. So they're not all monophyletic. It looks like Hysterionica is also not going to be monophyletic. So there's some questions about, best ways to do that. And I'm not a taxonomist, at least not yet. So I'm not going to be tackling these questions anytime soon. And the last thing I'll say here is this target capture probe set we built seems to work across a lot of the Asturiae taxa that we included. And then also we included chrysanthemum, and it sequenced pretty well as well. So maybe we could use that to start to, tease apart what this group should look like or should be named. And then, so this analysis is still running, the ancestral character state stuff, but I thought I'd plot all of our data on the tips, and then we can just kind of look at patterns that we're seeing pop up. So Erigeron, it looks like early on, diversified in the southern mountains and lowlands, and primarily these taxa are found in highlands of Mexico and central North America, kind of where Texas is. And then later on, diversified primarily in the temperate mountains, although it looks like maybe some diversification occurred in the boreal regions. And then there's this really interesting group here. There's 3 South American Andean taxa that I included, and they come out next to this Arctic taxon. And at first I thought I did something wrong. This seems odd, but after talking with some folks, they suggest I look at the actual species, and they look pretty much identical, maybe not identical, but very similar. And this pattern also is reflected in this really great paper from 1963, the amphitropical relationships in the floors of North and South America. There's been a recent update, but this one is really great because it describes all these taxa that have this kind of disjunction. And I'm not saying this bird, was the vector that carried seeds, but this bird does nest in the Arctic and then migrate down to the high mountains of the Andes, and does that every year. So maybe it was something like that allowed for this disjunction to happen. If we just look at the Alpine taxa, we find that it looks like the Alpine kind of biome occupancy evolved several times. It's not just one big clade of Alpine Arctic taxa. They're kind of spread out throughout the tree. That gives some credence to the lowland hypothesis here. But if we look at the Eurasian Alpine taxa, which I'm not including in this tree, they all come out next to this Arctic taxon, Erigeron eriocephalus. And so perhaps the Eurasian Alpine taxa are all descendant from this Arctic taxon. So that would be evidence for the Arctic hypothesis in Eurasia. And it seems kind of obvious when I say it out loud, but where a group originates might tell you more about how they've evolved into different biomes. Because I'm sure there are some Alpine taxa here that belong to groups that evolved in, say, the Alpine of the Himalayas, and they almost certainly came through the Arctic region to get here. And then finally, this is an odd thing we didn't expect. Erigeron glacialis is this pretty common subalpine plant that I see, every summer going up and down these mountains, and it pops into the Alpine, but we recover it as sister to the rest of Erigeron. And that kind of suggests maybe this group evolved pre-adapted to some of these environmental extremes. And so maybe it's not a surprise that we're seeing the Alpine biome evolve repeatedly in this group. Okay, so a lot of preliminary results, but some interesting stuff, I think. First, it seems like there have been several shifts into the Alpine, which would give some evidence to support the lowland hypothesis. There have been probably several Alpine to Alpine migration events. But there are probably more questions now than there were. So I'm become really interested in the role of hybridization in this group. We know it's happening, but is there an adaptive role as well? We've also been thinking about what the genomic or transcriptomic basis of this biome shift, what could have allowed that, or is it already kind of built into the genome architecture of this group? And then, of course, species boundaries, which we can't tackle with the data we have now, but maybe using a similar approach and broader sampling, we can start to tease apart what the species are and the names are. A lot of people to thank, but I just wanted to mention the herbaria. There's so many herbaria that I had to reach out to and talk with, and they were all so helpful, and none of this would have been possible without their help. And yeah, if there are questions, I'd be happy to answer any.
Very nice talk. Thank you. Mauricio, what is your question?
Yeah. Well, congratulations, Joseph. Yeah, amazing talk.
Thank you.
And really, I admire your courage to take on such a complex group like Erigeron. It's how you manage to key out things in Florida of North America inside Erigeron. It has been complex, right?
Yeah, I don't think I knew what I was getting into. I just thought, oh, this is a great group. And then I started and I thought, well, I got to keep going. So yeah, but it is a tricky group. Still learning every day. I'm learning something new. So yeah.
Absolutely. Well, very interesting that about the Niha and Histrionica and these other really odd things from South America getting inside a region. So I will be curious to see whether these larger clades inside what has been classified as a region have any morphological or other backing besides those very auspicious posterior probabilities that you have there, like that one that have the particularly backing on the shape of the leaves, but because putting all together inside a region will make the whole concept of region something rather difficult to define, right? Because
Yeah.
what is a region? So have you been looking into those clades? Any morphological backing at all, or?
I haven't looked deeply into it. I've looked at when I was collecting the samples, I would look and just make sure that the name matches what I'm supposed to be seeing. So I did that much. But as far as actually trying to like pull out characters that could define different groups, I haven't done a ton of that yet. And so actually, let me go back here. Yeah, you can see here, they look, to me, they look a lot like Erigeron just on their face, And so I think that's been a question for me too, is, what is Erigeron already, with this huge group, and so many of the characters are already so plastic that it's hard to say. This is a good one. One of the characteristics that, people say is Erigeron is equal or sub-equal filaries in 2, 3, 4 rows. But here, this columnaris, you can see they're pretty gradual. They're not quite what I would expect of an Erigeron. And if I saw this in the wild, I would think, no, I don't think that's Erigeron. And so, but there's so much plasticity even within Erigeron. So like this is at the base within the outgroup, but even, Canadensis was for a long time called Caniza because it has this kind of morphology. And so, yeah, that's just a long way of saying I haven't looked too much into the morphology of some of these groups, but already it's hard to, group anything because there's so much plasticity in this group.
But some of the things that you mentioned about the very broad share of biomes where these things occur across huge continent like North America extending into South America. Maybe that explains part of the situation. People working in different biomes and in a different continent wouldn't think that this thing growing in the lowlands it's in a steri, no, region maybe in the highlands, and maybe that's part of what explains this history of several genera being described for what essentially might be one highly diverse group. But then the question will be if we as taxonomists want to keep that as a huge thing that is hard to define, or we want to split it somehow. So yeah, this is a really cool project. Looking forward to
thank you.
See more on that.
Yeah, I'm
Thank you.
hoping that I can keep working on it, and appreciate your questions and comments. Yeah, Marissa.
Hi, thank you for your wonderful talk, Joseph. So I was curious, and I'm more familiar with glaciation in the eastern United States than the western one. But since a lot of these alpine kind of habitats often kind of act like islands nowadays because often they so far are like geographically apart nowadays, how could, do you know how connected some of these like habitats may have been if you did like niche modeling into like the last glacial maximum or something? Like, do you know like how connected some of these habitats might be for certain species?
Yeah, so the Alpine itself in the Southern Rocky, there's a couple papers on this that are really cool, but they show that in the Southern Rocky Mountains, the Alpine was pretty highly connected, but there was a
Mhm.
gap where I think it's called the Wyoming Basin in Wyoming, prevented continuous Alpine between the Southern and Middle Rocky Mountains. And that doesn't mean things weren't moving there, like things that are Alpine and subalpine were probably migrating across that. But so that was an Alpine kind of island pocket. The Sierras, I think, were pretty isolated as well. Like that's a big region, but it was isolated from other Alpine. Um, and then the Middle Rockies, until you get to kind of the panhandle of Idaho, that was all contiguous. But then the ice sheets came down around there and covered a lot of the Cascades and kind of the northern part of the Rockies. And so, if you look at this, taxa that are found in the boreal regions seem to also be found in the temperate, which suggests to me that a lot of these things were, migrating down and then migrating back up during the interglacials. And so
Okay,
yeah, that's my current understanding of that.
thank you.
Well, very nice. Yeah, thank you for a great talk, Joseph, and also to Ari for another great talk. So yeah, if there are no further questions, I guess we will see you all here in another month for some more nice talks.